reach the portal blood. And after that, they can be used by the animal for growth, health, and so on. But in between absorption and appearance in the blood, there is the step of the intestinal cells, and the metabolism here is very active. So I just placed here this little graph with all the metabolic pathways related to amino acids here in the small intestine. So the metabolism is very active. So that means that there is a difference between the dietary free amino acids that you find in the intestinal domain and the dietary free amino acids that you find then in the portal blood. And so with this difference, the question would be how important is this difference and how important intestinal metabolism is. And there have been studies in vivo that studied it, and they measure what they call net portal balance. So it's a bit complicated, but it's basically the percentage of dietary absorbed amino acid that you find back in the blood. So basically the share of dietary amino acids that you have in the blood after digestion. And we compiled some data from different in vivo studies and to look at what value we could find. And here you can find here the net portal balance here for the essential amino acids. And you can see that on average, you would find back 60% of dietary amino acids in the portal blood for the essential amino acids, but with a high variation in between studies, as you will see with the error bars. And if you look at now non-essential amino acids, you see that there is also a high variation for given amino acids, high standard, high error basically, but also here much more differences in between amino acids. With, for example, asparagine and glutamine almost not appearing in the blood at all after digestion. So basically, what we could conclude from this is that the variation is high in between amino acids for non-essential amino acids, but also for given amino acids in between studies. So we wanted to find to have a value, or say, okay, the intestine is going to use this percentage of amino acids and the rest will be found in the blood. But it's not the case. It's not possible to do that because there is too much variation in between studies. So why is there so much variation when measuring intestinal metabolism of amino acids? Well, of course, there are factors influencing like the composition of the diet, let's say about the composition of protein and amino acids, basically, but also obviously the fiber and how it interacts with the intestinal mucosa, a physiological status of the animal. Also, the fact that digestion is a dynamic process. So here I just illustrated with a little graph of the concentration of amino acids in the blood over time after a meal. And the thing is, like here we were looking at the figures I showed you before. It's just a given value. So X% of amino acid you find in the blood, but digestion it lasts several hours, and the process is not going linear as you see in the graph. So there is a peak in increase of concentration in blood, and then it decreases. And basically, it probably depends when you look at the use of nutrients by the intestine. It probably also depends on when do you look at it, at the beginning of the digestion, during the first minutes, during the last hours. So it's also a dynamic process. And finally, the experimental procedures are difficult to put in place for this. So it's a very invasive protocols with catheters, and even with these catheters, it's not really clear what is really measured, because in the end, you also measure what is used in the blood by other organs, such as the large intestine or the stomach. So you cannot really be very accurate and measure very accurately what's happening only in the small intestine. So here comes the interest of studying intestinal metabolism in vitro, and it has already been done by several authors. And the authors, what they use mainly isolated intestinal cells, and for mainly looking at things like substrate preference, so the use of different amino acids for energetic purposes by cells, or measure enzyme activity, and so on. But they use this only for static measurement, so not over time. And these cells are not used as a polarized laser, so they just incubate the cells in the medium with a molecule of interest, and they look at what they find back at the end. But there is not that polarized layer with apical side, basolateral side, and so on. So it's not really mimic well the intestinal barrier, let's say. So there are commercially available cell lines like the IPEG-G2 cell line. You see a picture here of a layer of these cells. And these cells have the advantage of being relatively easy to cultivate. It's not that easy, but commercial cell lines are relatively easy to cultivate. And they mimic well an intestinal layer because they form a layer and a polarized layer with an apical side and a basolateral side. And you can cultivate them on trans walls. And they are non-transformed jejunal cells from pigs. So, for example, there is a cell, famous intestinal cell model called CACO2, which are human cancer cells. And these ones, they are pig cells, and they are not cancer cells. So that's also an interest of these cells, and they are already quite used since like 10, 15 years, much less than CACO2, but they have a growing interest. On these cells have been used for measuring the effect of additives on the integrity of the intestinal barrier, to measure the absorption of different compounds and so on. But not really much to focus on metabolism in itself. So the question we were wondering would be then, can we use these IPEG-G2 cell line to mimic the intestinal metabolism of amino acid in vitro and to study metabolism? And to do that, so we aim to do an experiment or some experiments to follow the kinetic of carbon and nitrogen used by intestinal cells, by these intestinal cells, for two differentially metabolized amino acids. So we wanted to see how these cells, they metabolize the amino acids, but to see if they were really mimicking the metabolism like it is. We took two amino acids that have very different behavior. So we have first glutamine. That is a non-essential amino acid, and it is very much metabolized by intestinal cells because it's an important energy source for intestinal cells. So we know that in vivo, almost none of the glutamine appear in the blood because it's used for energy production by intestinal cells. And we use, on the contrary, we use lysine, which is an essential amino acid, and it's not much metabolized. It should not be catabolized in intestinal cells, and just a bit used for protein synthesis, but very low use. So that's what we wanted to do. Now a bit on the cell culture method. So we use the cells that we seeded in transwells. So see, if you're not familiar with what transwells are, so I put up here a photo of what it is and a little drawing here. So you have a well with medium on the basolateral side here, and the bottom, the paler color. You have medium on the top, the stronger color, let's say, the pinker color here. And so you have two medium, and you have a membrane that is porous here. And on that membrane, there is a layer of cells that grow. So the cells grow and they form a monolayer on that cell. And so the cells, they can use nutrients and transport nutrients from the apical side here, from the medium here, to the basolateral side here. So that's the interest of these transwells. And so that's how it looks like here. So it's on plates of 12 wells. And so the little plastic thing here is the transcell in itself. So with on the bottom, you don't see it very well, but on the bottom, there is that porous membrane here. So these plates we incubate for at 37 degrees. And we grow the cells there for 14 days before using them for our experiments. And we use a medium called DMMF12, so it's a commercial medium basically, but with porcine serum that we have in it. And just to monitor the cell culture, we measure an indicator that is called TEER, so it's transepithelial electrical resistance, every 3 to 4 days. And this is something we use to assess how the cells form a good layer and a permeable layer. So it's to evaluate the evolution of the cell layer over time that we measure that. So it's just an indicator, basically. So what do we do after 14 days of culture? So we take our cells on the transfers like this, and we use the labeled glutamine or lysine. So when I was talking about carbon and nitrogen a bit earlier, it's because we have labeled the carbon with 13C or nitrogen with 15N of the amino acids. And then we don't follow the fate of the amino acids by themselves, but the carbon or the nitrogen. So the interest with this is that we can really, in a nutrient-rich medium, we can really track what this carbon and nitrogen and find it. the thing is that if we measure the carbon and nitrogen, we don't really know if it's still on the amino acid or not. Although if, for example, the carbon and nitrogen do not have the same kinetic of use, we can then guess that probably they are not together anymore, so the amino acid might have been catabolized. And so when we use this, so we have to use them obviously glutamine or lysine separately, because we look at the carbon and nitrogen labeled. So if we mix them together, we cannot find from which amino acid it was. And as I was saying, so we wanted to have a dynamic approach, so measure at different time point over time, not just incubate and look after a certain time, but really measure over time what's happening. So at time 0, we place our glutamine and lysine labeled in the apical medium, like this. And then after different time points, we're gonna harvest the medium, either the apical medium, so the medium that is here on top of the cells, either the cells themselves, we're gonna harvest them, or the basolateral medium. On what we measure here, it's what is called delta. So it's basically to measure the amount of 13C or 15 nitrogen that has been that is present in the sample, and basically it's a kind of ratio, and it gives us an idea of the amount of carbon or nitrogen that we have in the sample. So it's a value that is a bit complicated to understand because it's a ratio based on the international standards, but as it is for now, we just mainly look at the kinetic and not the value by themselves. So for now, it's sufficient. And so where are we now? So very quickly, so it's a method that is in development. So we are working on this for now in the lab still, and I'm going to present you here the result from several trials that are mixed together. And I wanted to start with the question we were asking ourselves, because we are developing the method. So first of all, we're wondering if we can detect the enrichments, measure something basically in the cells that we harvest, because it's a very low amount of cells, as I will show you later. We were wondering if we would see variation over time of our enrichment, and if so, over which time period do the cells metabolize the amino acids. We were a bit also wanted to know what medium would be better to use to measure the amino acid use, and would we see differences in between glutamine and lysine as expected, and as we would expect for intestinal cells. So it's different question we were asking ourselves to develop the method. So first of all, a small thing about the transepithelial resistance. So I was saying, so it's electrical resistance that we measured over time to measure the integrity of the cell layer. And you can see here, so over 14 days, so that it starts very low, around 100, and it increases to more than 1,000, and then it stabilizes around 400. So it's a good value, because if it's too high, it's not physiological, it's not realistic. And if it's too low, basically, then the layer become very permeable, and then all nutrients go through. So that's a satisfying value. And so that's a measurement we use just to assess if our cells are growing well, basically. So then that was the case. So then the question was about, can we measure something in the harvested cells? So as I was saying, we are harvesting just the cell layer here to measure the amount of carbon and nitrogen that the label that we find in this layer. And in terms of mass, so it's just 0.7 milligram of dry weight. So it's very, very small sample. And we were wondering if we can even measure something in this and see something happening when doing our kinetics. So we did the first trial here with glutamine that was labeled on 3 different time points, and you can see here the time, so the measurement over time, so at 15 minutes, 60 minutes, and 120 minutes. And you see the enrichment for nitrogen here, and it's per 1,000. So the values by themselves do not mean much, but it's just more the kinetic. If it's above 0, it means that it's more than the natural enrichment, so that we find some of our labeled nitrogen in the samples. And you see that already after 15 minutes, we detect some nitrogen, from the glutamine in the cells. So that's incorporated very quickly. And that increases slowly over time. So it's difficult to describe the kinetic with only a few time points like this, but already that's encouraging. And it's a bit the same here, just for carbon. So the enrichment you see that follows the same kinetic. So we were already happy with this because we were able to detect something in the cells, and it's a very small sample, and still we see a variation over time as well. So we can measure something and see the increase over time. Now, then we were curious about what would happen in the medium over time. So we did first look here if we with the glutamine as well, label glutamine at the basolateral medium, so the medium that is underneath the cells, and here you can see over time. So that's different experiments, so the time points are not the same, but basically. Also, you see the increase in enrichment, so the nitrogen from the amino acids go through the layer, so it's absorbed, and already very quickly because we have points around above 0 after I think it was 1 minute here or 30 seconds. So that goes very quickly. Then we don't know if it's because it's actively transported or it's passive diffusion, but something we need to investigate a bit more. And we see a bit of similar trend for carbon here, although it's a bit slower in terms of increase over time, but also it's increasing slowly. And so we have a progressive increase, as I was saying, a bit faster for nitrogen. Would then it be linked with the fact that carbon from the carbon chain of glutamine is catabolized and you don't find it back because it's in the form of CO2? Well, it's not yet really possible to have a clear answer on this, but it's something we will investigate later on. Then if we look now at the apical medium, so the medium on the top of the cell layer where we placed our label glutamine at time 0, what's happening there over time? Well, here the results are a bit more surprising because if you look at here for the nitrogen, you see that the enrichment is stable or even maybe increasing. And that's something that surprised us. And you see also here that the scale is very different. So we are around 1,000. So it's very high. The enrichment is very high because that's where we placed our label. And one thing is probably that maybe the concentration is so high that we don't see much of the amino acid passing through. And so we don't see really the variation over time because it's a really tiny amount that goes through the cells. But the thing is, when we look at the arrangement like this, one thing to have in mind is that we look at the ratio. Here it's the ratio of 14 nitrogen to 15 nitrogen. And it is possible that if all the 14 nitrogen moves faster than our 15 nitrogen, then the ratio will increase while the nitrogen from glutamine is moving. But if it's moving slower than the rest, then we don't see really the result. So that's one thing with the labeled amino acid like this is to keep in mind that we are looking at a ratio. So that's probably also why we don't see really any evolution over time or even an increase. And it's the same with carbon. So probably that's linked with the fact that it's a ratio. And for example, if the 12 carbon from glucose is absorbed very quickly, then the ratio will increase, even though glutamine is also absorbed, but a bit slowly. So something we'd like to investigate over time. So we went here until 4 hours, and then until 4 hours, we don't see any evolution. So if maybe at first time points we see an evolution. So then with this absence of evolution of enrichment in the apical medium, we thought that maybe also that's the medium we use is too rich, because we have a medium that is called RPMI here that contains glucose, amino acid, vitamins, and so on, plus the glutamine we have, the label glutamine. But maybe it's so rich that the transporters are saturated, and so only a very small fraction of the glutamine is absorbed, and also because transporters for amino acids they can absorb different amino acids, so there can be competition in between amino acids. And maybe intestinal cells use more the glucose than the glutamine for their energy needs and so on. So we thought if we use a very poor medium that just contains a bit of glucose and salt plus our glutamine, we're gonna somehow force our cells to use the glutamine that they have in the medium and to use it a bit faster. And so we then looked again at the evolution of arrangement here in the apical medium, so on the top of the cells, to see if it will then drop instead of remaining stable over time. And actually, you can see here that just if you don't need to look at the absolute value, but just the trends, and you see that for the blue, the rich medium, or the green, the poor medium, well, the trend is a bit the same, so it's stable or increases slowly, and it's the same for carbon, so no really real evolution over time. It remains a bit stable. So whether we use the nutrient-rich or poor medium, we don't see differences in the apical medium. And I have other results with the cells and the basolateral medium that I won't present it here, but that suggests that probably the cells in the nutrient-poor medium are less active, and they didn't absorb much amino acids, probably because they were a bit stressed by this medium. But here we could not see any difference in terms of trend. The absolute value do not really matter here when you look at the arrangement. So then last question we were wondering here, so about the differences between lysine and glutamine. So we expect to see a difference because glutamine should be an important energy source for intestinal cells, while lysine should not be metabolized or catabolized by the cells. It just passes through the layer and appear on the other side. So I'm gonna just mix some results from different trials here. So so we used either here the label glutamine in orange or the labeled lysine. And yes, as I was saying, we're gonna look at the arrangement here in the cells. So if we look at first for the nitrogen, so you see that for the lysine in blue, it makes a line over time that's a bit linear with a linear increase. But you can also see that it increases already very quickly. Already in the first time point, you see that there is a bit of arrangement, so that's absorbed also quite quickly. And for the glutamine, it's more like the shape with a saturation, so that increases and then remains a bit stable after 2 hours or so. So the kinetic is a bit different here. And if you look at here for the 13C and the 13C, you see a similar kinetic in orange for the glutamine, so an increases and then it gets quite stable. While for the lysine, we see almost no increase or very little increase, so we don't find much of the carbon from lysine in the cells. So it's a surprising result. We do not really have a clear explanation for that yet. But we see differences in kinetics between the two amino acids, with a more linear increase for lysine and a more curvilinear plateau for glutamine. Now it's a bit difficult to draw a conclusion yet now on this, but something we need to investigate further. So we see differences, yes. And now to what are they due? It's something we need to investigate, and also something to see here that after like 30 seconds, we're already able to detect enrichment in the cells. So that would mean that already our cells contain the labeled amino acids. Is it the case, or is it because we didn't rinse properly our cell layer? We are not sure yet because it can go very quickly, but I'm not sure if it should be that fast. So just to conclude on this, so it's still a method we are developing, but we have already some interesting results. So we were able to measure the enrichment in the cells and to see an evolution over time. Similarly for the basolateral medium, so we see that there's an increase, a clear increase over time. So that these are interesting results. We need to dig in to go a bit further in the interpretation. However, in the apical medium, as I was saying, there is it's quite stable or increasing. So probably because of, as I was saying, the fact that enrichment is a ratio, so it depends a bit on the other nutrients as well. And I didn't show it here, but in the basolateral medium, we have measurements that were not so reproducible. So we had a lot of variation. So that's something we need to be careful on, because probably it's because sometimes the cell layer is very sensitive, and if it's slightly damaged, then that changes all the results we have. And so as I was saying, so we have more incorporation on carbon and nitrogen from glutamine in the cells over time than lysine. So interesting results, and we need to investigate a bit more on why we see this. So a bit of perspective now. So here I showed you only results in enrichment. So it's not, it's okay to look at the kinetic, but it's not that easy to understand and to interpret. And so we are transferring the result in milligram of 13C or 15 nitrogen. So basically to look at the amount of carbon we have at different time points. And then it would be, I guess, a bit clearer to understand. Okay, we put this amount at time 0, and then we find back this amount in the cells or in the basolateral medium. So it would be a bit clearer. But the arrangement is a bit it's a good measurement, but it's less intuitive, let's say. So we need to also to improve this problem. I didn't really expand on that, but the variation of the result and the reproducibility of the result in the basolateral medium. And go obviously a bit further, so we'll have to make some other experiments and repeat and to go a bit further in the carbon and nitrogen kinetics and the differences between lysine and glutamine and so on to have a clear picture of what we are looking at. And on the long term, it's data we would have liked to use in combination with modeling, so that a bit for later. So the idea was to combine a modeling approach and obtain the data to parameterize the model with the cell culture. And also something we had in mind was to go further into GCRMS. So here we look at just the amount of carbon or nitrogen we have in a sample, while we could look at the amount of carbon or nitrogen labeled that we have for example, for glutamine, for alanine, for glutamine, so in other molecules, to really be precise on saying, okay, we have the carbon from glutamine that is fine on this other amino acid or in urea or so on. So that could be a next step. Although the difficulty we have is that our samples are very, very small. So only a few milligrams and less than a milligram for the cells. So it's not that easy for the analytical part. So that's it for me.
Thank you, Clément. It was very nice presentation. For now, I would open the floor to the audience if they have some questions.
Hi, yes, I have a question. My name is Eliane van der Valk. I'm a research technician at Wageningen Livestock Research. So you shortly already mentioned that about the difference of transport, so whether it's either active transport or passive transport. Do you have an idea how you want to differentiate between whether what you see is active transport or passive transport?
We didn't do it yet, but there are some means, like you can block active transporters with certain molecules and then look at what goes through. That would be an idea. It's not something we have done, but I know that it's possible to block the transporters and then discriminate a bit between the active and passive at least. But yeah, it's not something we have done, and I don't know if we'll have time to do it.
Sorry, I had another question. So you also mentioned the permeability. Did you use some sort of measurement during the transport studies to measure for whether the integrity of your cell layer is fully intact? So whether there's already some permeability, just passive, with, for example, if it's dextran?
Yes, we did a bit that. We did that with we use also Lucifer yellow to measure the permeability of the cells in our conditions. And there was not much passive diffusion, so the integrity of the layer was good, because then we were expecting not much of that Lucifer yellow to go through, and that was the case. But then what the thing is, like, when we do the experiments, there are a few, let's say, movement rinsing from the layer, and that would be interesting to then measure that after, because we rinse the layer twice and then we use it. And the risk is always that that the layer is a bit damaged to touch it, or but even just with the change of medium to ice and so on, then the layer can be a bit damaged and maybe the permeability is a bit increased, and something we did not look at. But we did measure that with Lucifer Yellow, and the results were good.
Any other questions?
Jeremy Cottrell, calling from the University of Melbourne in Australia. Great talk. Thank you very much. It's very interesting. I had a question regarding the, I guess, following up from the previous question about passive transport, but also you mentioned the amount of washing that you do before you sample the tissue. Have you considered looking at L isomers, labeled L isomers with a different enrichment of carbon or nitrogen, so you could be able to discriminate between active and passive or unwashed samples?
So you said labeled what?
So you're using a 13C enrich, so you can use different isotopic labels or different levels of enrichment, but importantly, match that with an L isomer because you're using a D isomer of your lysine and glutamine.
Yeah.
So you use if you use a different isomer for your molecules and then partner that up with a different label, then you can discriminate between active and passive transport.
Yes, the thing is we didn't really. It's the fact that the transport was a big topic. How the amino acids are transported. We didn't really dive in that topic yet. We didn't really focus on this, how the amino acids are really transported, because we just wanted to see already if they were absorbed and used, and if we could find the back or mark or label and see if we had results around this where we're more focused on the fluxes by themselves. So for the transport, we didn't really investigate what are the possibilities, but I guess there are plenty of possibilities. But yeah, I don't know if I'll have time, unfortunately, to really focus deeper on this, because even about these cells, the IPIG2 cells, what transporter they express, and if they accurately match with what you could find in vivo, it's also something could be investigated. But something for now we just not ignore it, but we cannot really dive in it for now.
Thank you.
Any other questions? Okay, can I ask you a question, Clément?
Yeah, no problem.
So basically, you have 3 compartments that you checked. So the basolateral layer, medium, apical medium, and also the cells themselves. Yes. Can you comment a little bit about the fact that you didn't see big fluxes in terms of carbon and nitrogen between the apical and also the cells? Because if I understood your results correctly, so in the apical layer, I'm not sure if I'm saying it correct, but in the apical layer, you didn't see big fluxes, but in the cells themselves, you were able to observe some fluxes between the carbon and nitrogen. Can you comment about why this was the case and how could this happen biologically in a in vivo setting?
Yes, so the thing is, in the apical side where we put the label at first, so the enrichment is very high. It was around 1,000, 2,000. And when we are looking at the enrichment in the cells, we were around like 20, 30, 40, so much lower. So a variation, let's say that an increase in the cells might not mean a big change in the apical side. And so even though if it's a small amount of the proportion of 13C in the apical medium that goes in the cell, maybe that's a so tiny amount compared to the total amount of 13C in the apical medium that maybe we do not really see the change in the apical medium. So that's one thing. But obviously, the one big thing is that we are looking at a ratio. So if for example, the 12 carbon moves faster, as I was saying, than the 13 carbon, then maybe we have less 13C at the end of the experiment, but we don't see the difference because the 12C moved faster, so the ratio did increase, and so that's the tricky thing with looking at a ratio. And then you asked something about in vivo, Francis. If
Yes.
it was.
But actually, I wanted to ask you about your recent comment then. So it means that the main cells, so the cells that you used could have had less enrichment of natural isotopes. That's why we were able to see the delta compared to the apical membrane, because you probably have more 12 carbon and maybe natural enrichment of nitrogen there as well. Is that what you're saying?
Yeah. Well, the values were corrected for the natural enrichment, but it's more that we look at the movement on 13C and 12C, and so for example, if the glucose moves very fast, then probably a lot of 12C will move and disappear because the cells absorb the glucose and use it and catabolize it. And so then that means that we have less 12C. So then the enrichment in 13C, for example, in the apical medium, will increase, even though the quantity of 13C decreases also a bit, but slowly, because the cells use it a bit slower. So then you don't see a decrease in enrichment, but
Okay, interesting. Interesting. Okay. For the in vivo part, I'll ask you next time.
Okay.
So any other questions for Clément? Okay, then. I guess that's the last bit of questions that we'll
sorry,
have.
Junior.
Oh, yes. Go ahead.
Yeah, sorry. Just to ask out of, I'm curious to know, what is the most difficult part of controlling this kind of experiment?
It's an interesting question. Is the fact that we have we have some variability in between the wells in which we culture the cells. So because at each time point we have to sacrifice one well and use it to do the measurements. If we have too much variability in terms of permeability of the cell layer, in terms of how the cell layer differentiates and so on, then the results can be different, and so the variability of the well is a problem. And I think one thing I don't really work on this, but I have colleagues working on this, on characterizing these cells and knowing what cell types are there, do they produce or not mucus and so on. It's not really well known, so so probably that these kind of characteristics of the cells could explain part of the variation, but we do not really know that very well. So yeah, that variability can be in between different wells can be a problem.
Oh, thank you very much. It's quite interesting.
Okay, other questions? Okay, so I guess that's it. So again, thank you, Clément. It was a very interesting discussion. It was a very nice presentation. And for the next PIGWEB Junior webinar, take note of it in our LinkedIn page. So thank you and have a nice day, everyone.
Have a nice day.